All right, our next speaker is Harrison Tobi with Cornell Cooperative Extension, and he's going to talk about another aquatic species that's in the news a lot out here on the East End. Our, as he says, an iconic and commercially important Long Island shellfish species, the Peconic bay scallop. Welcome. So you just want to, that's forward. The bottom one is forward.
So
okay.
the opposite of what you think.
Yeah.
All right. Hi, everyone. Thank you for joining us today, and thank you for having me. So it's a really long title. So basically, I'll just kind of give you a summary of what I'll be going over today. I'm going to just give you a little bit of background on CCE's Bay Scallop Restoration Program, the history behind it, history of the Peconic bay scallop populations, their current status, and then some of our upcoming projects that we have currently underway and that will be underway for hopefully a couple more years to try to solve some of the issues that we are currently seeing. Right. All right, so this is important when we start talking later on about genetics. So there are two subspecies of bay scallops. There's the southern and the northern subspecies. So in Long Island, we have the northern bay scallop. Its native range is from Maine through North Carolina. Through that range, historically, it was a very profitable and sustainable fishery for the most part. It was important, obviously, to coastal economies and communities because it brought in a source of revenue to those economies and communities. Sorry about that. But then in the 1980s and 1990s, throughout that native range, there were a series of wild fishery collapses, and these have now been largely attributed to harmful algae blooms and habitat degradation. So we've got a few discussion topics. I'm not going to go through all of these right now. I just kind of want to highlight as to where we're going. So yeah, we're going to go over just our restoration program that we have at Cornell Cooperative. We basically do bay scallop aquaculture at Cornell. The hatchery is where it all begins. Our hatchery manager is Mike Patricio. He takes care of the algae culturing, which is fundamental to our process. So year-round, he is maintaining these algae cultures. Most of the time, during the winter, they're in probably 100 to 250 mil beakers. But right now, in that upper right-hand corner, you can see our C-cap system. All of those massive bags are filled with algae, which are going to provide food to our brood stock and both the larvae produced from that brood stock. So brood stock is just a fancy word of saying these will be the parents of the scallops we produce. This will also be important as we move further on into this presentation. Mike now has that brood stock. He's going to spawn them in about 4 to 6 weeks. They're going to remain in our hatchery for about a month. They'll go through the larval stage and the post-settlement stage, and then he'll keep them there until they're about a millimeter in shell height. And then we have an intermediate grow-out area in Cedar Creek and Southold. And Mike will take care of the scallops there for about 3 weeks. They'll hit about 10 millimeters. And then we'll move them out to our Orient Harbor Longline site. So if any of you, I'm sure a lot of you have driven out to Orient Point across the causeway. If you look off to your right-hand side, there are rows and rows and rows of buoys. So out there we have thousands of what are called lantern nets, which you see in that lower hand corner, which are being deployed off of our barge, the Shell Star. The scallops will remain out there for 3 to 16 months. The reason that there's variation is because we plant the scallops at different ages. So we like to keep some of the scallops out there for almost their entire lifespan. Scallops only live for about 18 to 24 months. Over that course, they typically spawn once or twice, so we like to keep a small subpopulation of spawner adults out there because it's very important for the scallops to be next to each other when they go to spawn. This helps increase cross-fertilization and helps for a healthy spawn. So the adult scallops, when they're out at our longline sanctuary, they're actually helping aid local recruitment. But we also plant those scallops at the end of the year, so baymen and recreational fishermen can go out and catch those adult scallops. Annually, we plant around 700,000 scallops a year. These will range from 3 months to 18-month-old scallops. The initiation of the program was in 2005, and since then, we've planted over 10 million scallops in the Peconic. We'll move on to our next discussion topic. This is another big part of the program. This is part of the program that I lead more specifically than the culturing. This is our bay scallop population monitoring. So our monitoring program is done via benthic dive surveys. These are conducted via scuba. We dive down, we document juvenile and adult bay scallop densities. We also document bottom type and bottom characteristics and changes over time. And then we also document predator type and predator prevalence as well. And these surveys are done either biannually or monthly. So there's two different types. So we do our biannual surveys in May and June. We're actually going to start our spring surveys next week. And then we do a set of fall surveys in October prior to the opening day of commercial harvesting. And we also do monthly surveys as well. This can range from 3 to 6 sites depending on where our field locations are. We always have 3 sites though, one in Orion Harbor, one in Northwest, and one in Cow Yard down in Flanders. For our biannual surveys though, we have 21 sites that range from Flanders all the way through Halleck. So, and now we'll move on to our recent and current field observations and data. So what are we seeing out there right now and in the last few years? So before we dive into that though, we're just going to kind of narrow our lens. So we just talked about the northern bay scallop, Peconic bay scallops are northern bay scallop, but specifically right now, just focusing on the Peconic bay scallop populations moving forward. Again, they had a massive fishery decline in the 1980s and 1990s. This was due to brown tide blooms, and this led to a near local extinction of bay scallops in the Peconic. In 2005, Dr. Tettelbach and Chris Smith founded Cornell's Bay Scallop Restoration Program, and in the subsequent years that follows, we saw a pretty big increase in the recovery of wild populations as well as the commercial fishery. So 2005, initiation of the program. We see that subsequent rise in commercial landings. So here we're looking at pounds of meat. This is going to be important because I'm going to show you another graph right after this. And then we're going to talk a lot about this. So in 2019, we had our first mass die-off year, and I'm going to go into quite a bit of detail as to what we now believe are the causes, and then after that, we're going to start talking about where we go from here. So this is similar to what we were just looking at. However, we have our survey data. So I was talking about the spring and fall surveys. This is data from our fall surveys, and it's important when we move forward looking at those die-off years. And then we also have commercial landings. So we were just looking at commercial landings in pounds. Now we're looking at it monetarily. And you can see 2019, our surveys go down, as well as the commercial value of the fishery. So what we can take away from this is that our surveys are a pretty good predictor of what we'll be seeing in commercial landings. And now we'll move on to more of our survey data. So 2019 was the first documented mass die-off year. As you can see, all these numbers represent a difference or a change in adult scallop densities between our spring surveys in May and our fall surveys in October. It doesn't really tell us when during that time, though, these mass die-offs occurred. That's why we do our monthly surveys, and I'll show you some data from that as well. Moving on. And then in 2020, again, you see a lot of massive declines in these different locations. In 2020, it was a 97% on average decline in adult bay scallops in our surveys. 94% decline in 2021. And then in 2022, we saw a 99% decline on average across our survey sites. So what are the likely causes of these mass die-offs? This has been a major question over the last few years. Recently, we published a few papers on this with our colleagues over at Stony Brook University, Marine Animal Disease Laboratory, as well as Dr. Gobler over at Southampton. So, big surprise, annual year-round temperature anomalies are now likely attributed to these mass die-offs. What we're looking at here is what's called a Z-score. We won't get too much into that, but blue and light blue represent cooler water temperatures. Red, obviously more elevated. And what I want you to focus on is 2019 to 2022. As you can see, there's a much higher prevalence of red. So what that means is that these years had much higher water temperatures compared to previous years, and these were the years that the mass die-offs occurred. So what is interesting, though, is looking at those winter months as well. So we like to primarily focus on like the summer months because we see a lot of elevated water temperature during those months, and we like to pay attention to that. But what is also happening is year-round elevated water temperature, and what this does is it increases the metabolism of the scallop when food supply is low. So they're burning energy over the winter when they normally wouldn't be, and those energy reserves will help them fight parasites and then those even more elevated summer water temperatures. So they're running out of energy over the winter when they're not getting energy back. And then in the summer, as water temperature goes up, its ability to hold oxygen goes down. So they're also experiencing lower DO in summer months as well, which is a massive concern. But one thing that is even more of a concern is a new parasite that showed up in 2019. This parasite has now been identified by Dr. Boston Malam's lab at the Marine Animal Disease Laboratory over at Stony Brook. It is a microparasite called Bay Scallop Microsporidia, or BSM for short. This was first documented in 2019, which was the first mass die-off year. It might seem like a smoking gun, but the thing is that they didn't test for it previous to the mass die-off year. It was only in that first year of the mass die-off that they sent these samples out for testing. It doesn't mean that it just showed up that year, so it could have been present before. But one thing to note is that it is very infectious. So it infects all the tissue, not just the adductor muscle, but it infects the kidney, which is where it is most detrimental because kidney function is very important for obvious reasons. It has a very high infection rate and intensity in summer months. So in that lower right-hand corner, we're actually looking at samples we provided to the Marine Animal Disease Laboratory in 2020. And as you can see, what I really want you to focus on here is that the intensity goes up between June through August. So the summer months when water temperature is highest, parasite intensity is highest. So we have the parasite, high summer water temperatures, and then we also have spawning stress. So when the scallops get ready to spawn, they allocate energy towards reproduction and away from basic life functions. So they're less able to mitigate the negative effects of this parasite and elevated water temperature during that time. And what we're looking at in this lower right-hand corner are 3 different sampling sites we have. And what I just want you to focus on are kind of like the peaks and valleys. So as the scallops get ready to spawn, what we're looking at is the gonadal indices of these scallops, which is just a metric of how reproductively active they are. So they ramp up, and then that drop we kind of take as a signal that they've spawned out. And if you see when they're spawning, it's right around the month of July. And then in the lower left-hand corner, we have 2021 and 2022 field data from those monthly dive surveys. And what we're seeing here is that by August, almost 100% of the adult scallops in our surveys have disappeared. So again, high water temperature, parasites, spawning stress, massive mortality in those summer months. So we now have a pretty good idea of what's causing these mass die-offs, and it seems like doom and gloom, and that there's not a whole lot of potential to bring this fishery back. However, now that we kind of understand what's causing it, we can now take that information and look for solutions, which is very important. So at Cornell, we have 2 new and upcoming projects. These are actually currently underway. Both of these projects started last year. This year is just going to be the field deployment of these projects. So the first project is our fall spawn versus summer spawn scallop project. And then our next one is our restorative breeding program. So this restorative breeding program project is a massive project. It's a collaborative project, again, with Dr. Alam at SUNY Stony Brook. And we'll get into more detail on that in just a second. So we just saw this graph looking at the reproductive activity of our scallops at our sampling sites. We focused on the summer spawn. Scallops can also spawn in the fall. In the previous presentation, we talked about bed hedging when it comes to spawning. So scallops, we don't really understand the causes of these fall spawns or what role they play in population dynamics, but they do occur year to year. They're not as productive as a summer spawn, but they do happen and they can be important. Just kind of to give you an overview of what a fall spawn scallop looks like. So scallops live for 2 years. In that first winter, they get what is known as their annual growth ring. And what we're looking at here, on the left-hand side, you see a scallop from a spring or summer spawn, one of those July spawns we were just talking about. So they have a much longer time to grow prior to that first winter. So for a summer or spring spawn scallop, you're going to get a scallop that's about 50 millimeters in shell height starting that second year. Whereas a fall spawn scallop, we call them nub scallops. So they're born in September or October. They have a much shorter growing period. They have a much smaller annual growth ring. Kind of when you're thinking about a growth ring, think like a tree. When you cut it in half, you see those concentric rings. Growth stops during the winter. You get a very, very identifiable ring. So why do we care about fall spawns? Well, as we were just discussing, majority of the adult scallops are dying in those summer months. So we might be losing robust fall spawns, and we might be losing any potential population benefits that come with them. Additionally, we still don't really understand, like I was saying, the role these fall spawns play in overall population dynamics. So not only may we lose them, we might lose our ability as a scientific community to get a better understanding of what role they play. But I think just kind of, this is anecdotally, but a good example of the importance that they might play were the 2 years prior to the mass die-off. So earlier I was talking about the declines in adult bay scallops between our May and our October surveys. In 2017 and 2019, the 2 years prior to the mass die-off, we actually saw an increase in adult scallops. And this might sound weird because they only live for 2 years. How can you have an increase in adults in that second year? Well, Dr. Tettelbach hypothesized that the reason we saw this increase is because I just showed you those shells. So a fall spawn scallop in the spring might be 5 millimeters in shell height, whereas a spring or summer spawn one will be like 50 millimeters. So it's much easier when you're out there diving to see a 50 millimeter scallop. I was just diving earlier this week, and at one of our sites, visibility is like 2 to 3 feet. So you have, poor visibility combined with small shell height in the spring. Steve thinks that this might be why he saw these increases in 2017 and 2018. So in 2017, we saw an increase as high as 943%, and then in 2018, as high as 958%. So this increase and the presence of these fall spawn scallops could indicate that they do play an important role, and we should try to figure out, is this something that we need to look into more? So what we're doing this summer, I actually just went and set up these field sites earlier this week. What we're going to do is a comparative field study. So we're going to take scallops from our June spawn last year and scallops from our October spawn last year. We're going to put them in aquaculture gear. And then through May through October of this year, we're going to track their survival. And then we're also going to track their parasite intensity. So the benefits of fall spawning, we already talked about, trying to bring back that robust fall spawn, and we also talked about trying to get a better understanding of what role it plays. But when we're looking at it in regards to the parasite, so the parasite is most prevalent in the water and in the sediment and in the scallops during those summer months from June through more or less mid-August. So if you're a scallop and you're born in June or July, you have a much larger window of infection and exposure to this parasite compared to if you're born in September or October. So a scallop born in September or October might not even get infected in that first year. Fast forward through the winter, come the next spring, a fall spawn scallop might be more or less naive compared to a scallop born in June or July. So a lower parasite load in that second year, they might have higher fitness. This higher fitness might allow them to better combat the stressors of elevated water temperature as well as that initial spawning stress, and they might be able to survive better to that fall in the second year and contribute to fall spawns, but also, more importantly, contribute to commercial harvests. Our second project we have going on is a restorative breeding project. This is does it look like that to you guys too? Okay, I don't know what is going on there, but bear with me. So again, Dr. Bossum Alam, Marine Animal Disease Laboratory at Stony Brook. This is actually using genetics and parasitology. So in January of this year, Bossum actually sent out 900 samples to have genetics done. These samples range from Maine down through New Jersey. He actually got samples from 1903 from the Natural History Museum. And the idea behind sending out this massive set of samples is to get an idea of genetically unique populations within that range. Majority of the samples came from Long Island, and the reason majority came from Long Island is because we're trying to identify genetics that might increase survival. So what we're looking at in the right-hand corner, the best one to probably focus on is the upper right. Each dot represents an individual scallop. Each color represents a different sampling time. As you move from left to right, those are different dominant genes. So our earlier samplings, a lot of those scallops had very similar genetics. We had about 60% survival at this site, so 40% mortality. So as you move through the summer, the weaker scallops, presumably, have died out, and then by the fall, which is represented in yellow, you have a different dominant genetic makeup. And what we're assuming is that those yellow dots, the genes represented there, are genetics that promoted the survival of those scallops. So another thing that needs to be emphasized here is that all these scallops came from the same parents. So they share the same parents, they're the same siblings. So what we want to do, or what we have done, is we've taken the scallops represented in yellow, we use them as brood stock, and then we're going to take the scallops from that brood stock this summer, and we're going to put them out at different field sites, and then track their survival and track their parasite intensity, and see if there are increased survival based on genetics. Again, we just kind of covered this. Where are we going with the restorative breeding project? We collected those year 2 adults last year from high mortality areas. We bred them. We're hoping that their offspring will get those genetics, and that those adult scallops had genetics that promoted survival. This summer, we'll take those the progeny from those spawns, deploy them at our different field sites, track progression of the parasite, and also track survival. Additionally, we do all these spring and fall surveys. Any adult scallops we find, we're also going to give to Dr. Alam to send out for genetic testing as well. So in summary, our initiation of the Bay Scallop Restoration Program was massively successful at bringing back the fishery, massive increase in commercial landings and economic value. But again, since 2019, we've been experiencing these mass die-off years. So massive concern. This doesn't just affect, obviously, the natural populations, but we're also seeing that reduction in the commercial landings. So it's an economic and ecological issue. However, with these new projects we have coming up, we're hoping that we can take our findings and develop adaptive management strategies. The fall spawns could decrease that window of exposure to the parasite in the first year. But I really think the restorative breeding project is going to be a massive, massive success. It's going to take years, likely, to really hone in what is driving resilience and if resilience can be selected for in our spawning process. Last but not least, I'd like to acknowledge my coworkers over at Cornell Cooperative, the Peconic Estuary Partnership, NRAC, NSF, Robbins Island, or More Charitable Foundation, my colleagues over at Stony Brook University, Dr. Alam's lab is amazing to work with. You know, we do all the field work. He does all the technical lab work, so it's a very synergistic relationship. I get to be out on the water all summer. He gets to work in his lab. And then I'd also really like to thank Suffolk County. They have been a long supporter of this project. They helped start they were the initial funders of the project. I actually presented to them on Monday, and they were very excited to see where this is going. They're massively supportive of it. So, again, we really, really appreciate Suffolk County's support. And on that, I will take any questions. So it was a difference of 4 degrees. Good question, though. Did everyone hear that question? So he was asking what the colors on the temperature graph represented, what the difference was. So it was a difference of 4 degrees overall, 4-degree temperature difference. Truly. It's an epicomplexan parasite. And the reason I mentioned adductor mussels is because it's very similar to the parasite that causes gray meat disease in the Atlantic sea scallop fishery. So earlier, the presentation before me was talking about fisheries observers. I was actually out on fishery boats back when the gray meat disease was actually first really becoming a problem. So it's a microparasite in the phylogeny of epicomplexan, or coccidium. Yeah, so that graph we were looking at was looking at only sample tissues from scallops. Oh, no, we have a question.
Yeah. In addition to, thank you for the presentation and all the hard work that you guys have been doing on this topic. In addition to the selective breeding projects that you've mentioned, have you guys been exploring or concepting the idea of looking further south for brood stock that might be more adaptive to them?
So, we have thought about that. Currently, we're trying to preserve. The reason we're doing that massive genetic study is because we want to identify genetically, is the Long Island, are Long Island bay scallops genetically unique, or are they similar to ones found in the South? If we can show that they're similar, then maybe we can start taking scallops from further south. But as of right now, we're trying to protect the genetic integrity, if you will, of Long Island populations. So that's really the main goal right now. That's why I brought up northern and southern subspecies. For right now, we want to focus on preserving Long Island genetics as much as we can. And I also think I think DEC might have regulations against that currently right now, but I'm not positive on that.